Auzubillahiminash-shaytanir-rajeem. Bismillahir-Rahmanir-Rahim. My name is Aftab Aleem Farooqi. I am Assistant Professor in Biochemistry at Govt Degree Science College, Malir, Karachi. Now I am starting the lecture for B.Sc class in subject Biochemistry, which is continued lecture number 2. And I am continuing the chapter Metabolism, Basic Concepts and Design. And here I am continuing the energy cycles, and here the energy cycles which we have to read in B.Sc third year, and the reference book for this topic study is Harper's Physiological Chemistry 23rd edition. So I am starting the chapter. The topic is the basic metabolic pathways process the major products of digestion. So here you have to understand that after complete metabolism of macrobiomolecules like carbohydrate, proteins and lipids, you will yield the simpler one molecule glucose will be obtained after the complete digestion of carbohydrate. Amino acid residues will be obtained from the complete digestion of proteins, and fatty acids and glycerol will be yield after complete digestion of lipids. So these simpler one molecules will undergo into the further processing means they will further replenish in order to produce the ATP molecule through the energy cycles. So mammals such as humans need process the absorbed products of digestion of dietary carbohydrates, lipids and protein. These are, I have already told you, glucose, fatty acids and glycerol and amino acids are respectively obtained. All these products of digestion are processed by the respective metabolic pathways to a complete process, or by the respective metabolic pathways to a common product, that is acetyl coenzyme, which is then ultimately oxidized by citric acid cycle. Now, as you have understand that after complete digestion and absorption, carbohydrate yields simple glucose molecules, protein yields amino acids, and fats produces fatty acids and glycerol. Now these three simpler one molecules will undergo into the catabolic process, catabolic pathways, and they will, all these three components simpler one molecules will produce the main one common molecule, which is produce the acetyl coenzyme A. So question is in the mind that simple sugar glucose, how will produce acetyl coenzyme A? You know that glucose is catabolized through the glycolytic pathway aerobic phase. Glucose will undergo into the will goes into the aerobic phase of glycolytic pathway, and then glucose is converted into pyruvic acid. Then this one glucose molecule will produce the 2 pyruvate or pyruvic acid. These 2 molecules will enter into the citric acid cycle by conversion into the acetyl coenzyme. Therefore, pyruvic acid will converted into the acetyl coenzyme A. This acetyl coenzyme A is responsible for the initiating of citric acid cycle or Krebs cycle. This is a much producer energy cycle, in an energy producing cycle, which produces a much more quantity of 36 ATP molecules production as a result of the 2 as pyruvic acid will convert to the acetyl coenzyme A, and acetyl coenzyme A will lead to 36 ATP molecules production. Therefore, one complete glucose molecule will be catabolized through the aerobic phase of glycolysis. Therefore, 36 ATP molecule will be produced. Now, the amino acids will catabolize. So, amino acids, how they are catabolized? Because you know that amino acids will undergo into the deamination process. Therefore, the deamination process, they will be catabolized, undergone into the non-protein nitrogenous substances, that is the urea molecule. And as far as they are converted into the acetyl coenzyme, they can also convert it into the acetyl coenzyme. This acetyl coenzyme will enter into the citric acid cycle, and produces the ATP molecule production. Now the third one is the fatty acids and glycerol. These fatty acids and glycerol are yield as a result of the lipid metabolism, and these lipid metabolism, then this fatty acids will undergone into the catabolic pathway. Therefore, it converted into the acetyl coenzyme. This acetyl coenzyme will enter into the citric cycle and again production of ATP molecule, and also the carbon dioxide gas are also produced. So this is the main digestion, absorption and energy producing molecules obtained. Now carbohydrate metabolism is centered on the provision and fate of glucose. Now glucose is metabolized to pyruvate and lactate in all mammalian cells by the pathway of glycolysis. So you have understand in which condition glucose is metabolized to pyruvate. When there is oxygen gas is available, therefore glucose will be oxidized in aerobic phase of glycolysis. Therefore glucose molecule is converted to the pyruvate. Therefore this pyruvate is converted again into the acetyl coenzyme. This acetyl coenzyme will enter into the citric acid cycle by combination with the oxaloacetate, then form the 6 carbon atom containing citric acid molecule. This citric acid molecule is responsible for the initiating of citric acid cycle. Okay. Now in the second part, this is the lactate is also produced from the glucose. In which condition lactate is produced? So glucose is a unique substrate because glycolysis can occur in the absence of oxygen when the end product is lactate. So this is very important that when the oxygen gas is not available for the glycolysis process, therefore glucose will undergo into the anaerobic phase of glycolysis, and resultantly glucose will be converted into the lactate, never in pyruvate. Why? Because reason is that in muscle hexokinase is secreted, never glucokinase. That's why the reason glucose is converted into the lactate, neither in pyruvate. And also important point is that when animal tissues cannot be supplied with pyruvate with the sufficient oxygen to support aerobic oxidation of the pyruvate, क्या होता है कि जब एनिमल टिश्यूज को सफिशिएंट ऑक्सीजन अवेलेबल नहीं होती है टू सपोर्ट एरोबिक ऑक्सीडेशन ऑफ पायरुवेट, so NADH produced in glycolysis. Okay, so this in which condition also there is another condition in which the anaerobic phase of glycolysis occur when in strenuous muscle contraction condition, in heavy physical body activities like athletes or when they are jumping or running in the event. In that case oxygen gas potential is very decreased, become very decreased in the human body. So in that case human body needs sufficient energy, rapid sufficient energy for the body activity. So in that case the anaerobic phase of glycolysis, the glucose is converted into the lactate, never in pyruvate, because of the absence of oxygen gas. And also NADH produced in anaerobic phase of glycolysis. So NAD is regenerated from NADH by the reduction of pyruvate to lactate. तो यहाँ क्या होता है कि जो पायरुवेट की रिडक्शन नहीं होती है फ्रॉम यानी NADH से जो होता है बाय द पायरुवेट टू लैक्टेट बनता है, तो यहाँ पर क्या होता है कि वो प्रोड्यूशन नहीं होती है. तो certain other tissues and cell types, retina, brain, erythrocytes also produce lactate from glucose under aerobic conditions. So lactate is a major product of erythrocyte metabolism in RBCs. The reduction of pyruvate is catalyzed by lactate dehydrogenase, which forms the diisomer of lactic acid at pH 7. So the understand is clear that in which condition this is produced lactate. And also I have earlier told you that aerobic phase of glycolysis will yield the acetyl coenzyme through pyruvate and then enter into the complete oxidation through the citric acid cycle and produces the 36 molecule of ATP production, and carbon dioxide gas will be releases. So these ATP are produced in oxidative phosphorylation. So glucose is a major fuel of many tissues. Now look here that glucose is converted into the glycogen. So in which condition glucose is converted into the glycogen? Polysaccharide biomolecule. When there is sufficient or excess synthesis secretion of glucose molecule will be obtained after result of the carbohydrate metabolism, and these glucose molecules are not needed for body function or body vital activities. So this glucose will be converted into the glycogen in the liver through the glycogenesis process, and this glycogen will be stored in the liver. So this is stored in skeletal muscle and liver. So skeletal muscles in glycogen is very least in quantity stored, while in the liver the major quantity of glucose is stored in the form of glycogen in the liver. Now second pathway is pentose phosphate pathway. This pentose phosphate pathway arises from intermediates of glycolysis. Here is an important point I would like to explain here, that glucose consists of 6 carbon atoms, while pentose type of sugar, that is ribose, which consists of 5 carbon atoms. So in this pathway 1 carbon becomes lesser, will be reduced, and 6 carbon atom glucose molecule will be lesser and converted into the 5 carbon atom containing ribose sugar. So in this process pentose phosphate pathway, as I will told you, intermediates of glycolysis है, वहाँ से ये बाहर आता है और pentose phosphate pathway ribose sugar synthesize होती है. It is source of reducing equivalents to hydrogen atom required in the synthesis of fatty acid. देखें वहाँ जब 6 कार्बन एटम से 5 कार्बन वाली शुगर राइबोज यील्ड होती है तो वहाँ से 2 हाइड्रोजन बाहर आते हैं, और these hydrogen 2 hydrogen reducing equivalents are required in the synthesis of fatty acids. Okay, so it is also the source of ribose sugar that is important for the synthesis of nucleic acid and nucleotide metabolism. Now here the amino acid metabolism involves transamination. Another type of the in simpler burn energy producing molecule amino acid, this is obtained as a result of the protein metabolism. So here amino acid will further undergoes into the metabolic process, especially in the transamination where amino group will be removed. So amino acids are necessary for protein synthesis, you know very well, and some must be supplied specifically in the diet. Okay, so the essential amino acids. So here I would like to explain an important point that there are 2 types of amino acids. One is essential amino acids, another are non-essential amino acids. Essential amino acids are those amino acids which are not synthesized in human body and they must be required in our balanced diet, while the non-essential essential amino acids which are already synthesized in human body. So the remainder or non-essential amino acids are also supplied in the diet, but they can also be formed from the intermediates of transamination using the amino nitrogen from other surplus amino acids. तो अब यहाँ पर क्या होता है कि जो दूसरे surplus amino acids हैं वहाँ पर से amino acids जो intermediates हैं वहाँ पर amino nitrogen उसकी transamination हो जाती है, तो इसके नतीजे में क्या होता है surplus दूसरे amino acids से यहाँ पर इनकी synthesis होती है amino acids की. और साथ ही यहाँ पर क्या होता है कि after deamination यहाँ पर जब अमाइनो ग्रुप की रिमूवल होती है तो एक्सेस अमाइनो नाइट्रोजन इज रिमूव एज यूरिया. सो हियर इंपॉर्टेंट पॉइंट इज दैट द यूरिया इज अ नॉन प्रोटीन नाइट्रोजनस सब्सटेंस. सो अमाइनो एसिड विल अंडरगोज इनटू द कैटाबोलिक प्रोसेस, कैटाबोलिज्म ऑफ द अमाइनो एसिड. सो जब कैटाबोलिज्म ऑफ अमाइनो एसिड्स होते हैं तो यहाँ पर जो है रिजल्ट ऑफ द नॉन प्रोटीन नाइट्रोजनस सब्सटेंसेस प्रोड्यूस होते हैं, जैसे कि यूरिया, अमोनिया, यूरिक एसिड, केटाइन, केटिन, न्यूक्लियोटाइड्स एक्सेट्रा. तो यहाँ पर जो है आफ्टर डीएमाइनेशन एक्सेस अमाइनो नाइट्रोजन वो रिमूव होती है अमाइनो एसिड से, और इन द फॉर्म ऑफ यूरिया एज द कार्बन स्केलेटन्स दैट रिमेन्स आफ्टर. So the transamination will also yield another important points. I would like to explain that transamination also oxidize to carbon dioxide by the acetic cycle. देखिए अब यहाँ पर एक इम्पॉर्टेंट पॉइंट ये है कि जो पहला पॉइंट है ट्रांसअमाइनेशन, जब अमाइनो एसिड की ट्रांसअमाइनेशन होती है उसमें से अमाइनो ग्रुप की रिमूवल होती है, तो यहाँ पर अमाइनो एसिड ऑक्सीडाइज हो जाता है एसिटाइल कोएंजाइम में, एसिटाइल कोएंजाइम मॉलिक्यूल में. दिस एसिटाइल कोएंजाइम मॉलिक्यूल विल एंटर इनटू द citric acid cycle production, in citric acid cycle energy cycle, which is responsible for the much energy ATP molecule. And as far as the amino acids also undergone into the glucose formation through the gluconeogenesis process. What is the gluconeogenesis process? Gluconeogenesis is a process in which non-carbohydrate substances like fatty acids, amino acids, lactic acid are converted into the glucose. So therefore there is another important chance for the amino acid is that they also so the transamination process remove the amino group, they can also convert it into the glucose molecule. As far as the transamination is also formation of the ketone bodies. Amino acids are also responsible for the ketone bodies production. So look here the diagram. Diet protein which produces after complete digestion of protein, amino acids are produced. These amino acids will enter into the left side arrow again in the formation of tissue protein that resulted formation of the muscles. As far as in the right side, the amino acid undergoes into the catabolism process. They will produce the non-protein nitrogen derivatives, that is the urea, ammonia, uric acid, etc. As far as the transamination will undergo into the different processes like the transamination removal of the amino molecule from amino group, that will lead to the glucose formation of the glucose from the glucose molecule. So the gluconeogenesis process. As far as the transamination is also responsible for the amino nitrogen in glutamate. तो यहाँ पर क्या होता है कि अमाइनो नाइट्रोजन जो है ग्लूटामेट अमाइनो एसिड्स में उसकी वो नाइट्रोजन रिमूवल हो जाती है, डीएमाइनेशन. तो डीएमाइनेशन से क्या होता है कि अमोनिया ग्रुप रिमूव होता है. ये अमोनिया ग्रुप जो है ऑक्सीडाइज होकर यूरिया में कन्वर्ट हो जाता है. उसके बाद यहाँ पर ट्रांसअमाइनेशन अमाइनो एसिड की दोबारा होगी, तो एक और मॉलिक्यूल वही जो मैंने बताया आपको शुरू में कि ट्रांसअमाइनेशन से वहाँ पर जो अमाइनो एसिड जो है वो उसे एसिटाइल कोएंजाइम प्रोड्यूस होता है, दैट विल लीड टू द सिट्रिक एसिड साइकिल एंड गोज इनटू द रिमूवल ऑफ द 2 कार्बन डाइऑक्साइड मॉलिक्यूल एंड 36 एनर्जी मॉलिक्यूल एटीपी प्रोडक्शन. As far as the transamination will yield the ketone bodies production. So the amino acids are responsible for the production of ketone bodies. So this is the simpler one explanation, very simplest explanation of the energy cycles of the simpler one molecules of their respective macrobiomolecules in order to produce the energy cycle. So here we have completed the brief introduction of the energy cycles in different metabolic or catabolic pathways of these simpler one molecules. So we have completed this chapter, and again we will start the next chapter is electron transport chain. Okay students, thanks.